Okay, so good morning, good afternoon, everyone. Warm welcome to the first of this year's Target 2035 webinars. So my name is Andrew Leach, I'm at EBI. I'm going to be moderating today's session. So we've got six great talks lined up for you today. And they're broken into two sort of blocks of three talks. Just a couple of housekeeping matters so that everybody's aware how we're planning to run these. I know many of you will have dialed in previously, so you'll be quite familiar with the way that these operate. First thing to note is that we will be recording them. The recordings will be made available from the target2035.net website after the event. So do encourage your colleagues, if they can't make it today, to go along and take a look. We have, as I mentioned, the two sort of sections, and we will run the talks consecutively without a break because we have 10 or 15 minutes allocated at the end for sort of verbal Q&A. But what I would encourage everybody to do, if they have a question, as soon as you formulate that, please enter it into the Q&A box at the bottom right of the Zoom window. And when the speakers are finished, then if they can, they will endeavour to type in their answers to those questions. But clearly some questions are a bit more difficult to answer easily in a sort of textual way. So we may keep some of those over for the verbal Q&A. And of course, that can continue through the session as well. And then just finally, by introduction to the overall programme, as you can see from the speakers and their titles, I think we have a great mix of data informatics, data integration, computational methods, simulation, and of course, artificial intelligence-based methods. And so I'm going to hand over now, with no further ado, to our first speaker, Tudor Perea from the University of Mexico, who's going to be talking about the druggable genome and target classes.
Yes, hello. Hope you all can hear me. Good morning, good afternoon, everyone. Thank you for inviting me in this exciting webinar series. I'm going to talk to you about the understudy genome from an Idg perspective. And the whole story started when Al Edwards and Gary Bader and colleagues came up with this observation that 10 years after accomplishing the Human Genome Project, 75% of the protein research was still carried out on 10% of the known genes. And that sort of caught the attention of program officers at NIH, and they launched the Illuminating the Drugable Genome program. And part of this IDG program, we often ask the questions as to where we could use informatics, data science, and machine learning, what some people call AI these days. And you can pretty much say that whether you're looking at diseases, so we had recently a paper counting the number of rare diseases and looking at therapeutic modalities. So you can use, of course, AI and informatics in EMR processing, nosology, ontology, etc. And then, of course, in targets, which is what this project is about, the most critical part is identification and validation of novel targets. So that remains probably the holy grail in drug discovery as of today. And then, of course, in the drug areas, virtual screening methods and a lot of in silico methods have been used quite a lot. And some of us who are on this call have participated in this effort to curate more than almost 1,000 drug targets out of the human genome plus pathogens for about 1,600 approved drugs. And just because I was curious about this, about 2/3 of those drugs are orally formulated. You can find more information in Drug Central, and those of you who are interested in the slides, they will no doubt be made available, so you can follow that link. And this is the makeup of the IDG program, except for the R03 funded projects. There's too many of them to list on a single slide. By my count, there's about 32 that have been funded, but essentially there's a large set of projects that are dedicated to gene-protein coupled receptors, and you will hear from Brian Charquet later. Ion channels and kinases. So these three large groups generate experimental data. There is an outreach component, which is led by Stefan Schürer, and there's 2 knowledge management centers, plus a number of tools that are being developed by a variety of groups, including the Reactome group, for example. So essentially what we do for the KMC, which is the Knowledge Management Center, we take a lot of the information that's been made available, and we infer that information. We take data from genome and protein information. There's the Harmonisome project from Avi Mayan. We currently incorporate a string data set for protein-protein interactions. We, of course, have to utilize a number of ontologies, whether it's for tissue localization or diseases, etc. And then, of course, a number of chemocentric databases, including Chembo. And then all that information gets into Target Central, and then it gets labeled into TCLIN. So TCLIN are approved drug targets. TChem are targets for which known small molecules are available. TBio are those for which you understand the biology, and then TDark are proteins which are understudied. And a number of criteria that discuss these have been published in Nature Drug Discovery. So we take all that data and we parse it through a portal called Pharos, and you can access Pharos at pharos.nih.gov. And of course, there is an ongoing process of target evaluation, and we have target prioritization lists. We work, for example, with International Mouse Phenotype Consortium, as well as internally with our own DRGCs. All that information gets targeted, visualized, and of course would be subject to machine learning. So what kind of data goes in? We recently had a paper published in Nucleic Acids Research. We take a lot of information, and I put here a little bit. So for example, with Shore Campbell, there's an ongoing project led by Adri Leach, where biochemical bioactivity data from patents gets indexed to small molecules. So we try to capture literature in addition to the typical JMedChem type publications. There's a drug target ontology that was developed by Stefan Schürer. And then, of course, the Harmonisome, which I already mentioned. It basically takes experimental data from about 100+ data sets and collates it into a single matrix that tries to give us ideas as to how much do we really know about proteins. There's a number of other common fund projects that we take into account and index. GTEx, LIGS, and the IMPC are such examples. And as you can see from this list, it's quite a variety of types of data. And just a brief summary statistics, more than 700,000 target attributes and more than 57,000 disease phenotype attributes, plus more than 500,000 ligand attributes. And you can go to Pharos and find out more about it. So if you were to go to Pharos, this would be the entry page. The most developed part is on targets, but of course, we are making progress on diseases and ligands as well. So I thought I would give you a feel as to why this is such a complicated problem. We work closely with Lars Jensen in Denmark, and this is a screenshot to illustrate the type of data that are available in the diseases database. This database is updated on a weekly basis, so it's quite current as far as indexing data from PubMed goes. In 2015, when it was launched, it was based on purely on text mining data from about 25 million patents. It now has the capability, and it's been developed to process data from more than 31 million abstracts, but also the part of PubMed Central that allows complete download. So not all of the papers in PubMed Central are available for download, and hence there's a distinction there. But it's really reinforcing the ability to text mine and map the association of diseases and proteins/genes. One thing that's intriguing is that depending on where in the text that association is found, diseases will give it a different score. And part of the novel integration of diseases is that it now uses BioBERT, which is a bidirectional encoder for text mining that essentially allows us to machine learn whether a gene name is truly a gene name or a false negative in the context of where it was found. And that pretty much gives us one of the strong channels in Pharos. We currently have 11 different channels when it comes to protein disease associations. So it's really complex, and it's quite a challenge to harmonize this, partly because a lot of the literature is single gene centric, where in fact we know that some of these targets form complexes, which in turn become the drug targets. So for example, in type 2 diabetes, this sulfonylurea 1 receptor is a combination of a potassium channel and a ATP binding cassette transporter. So the real challenge when you run machine learning and artificial intelligence is, do you know that these associations exist between the targets, and do you take that into account when you run your predictions? So we've developed a workflow in machine learning, and essentially we take all the data. So all of the data in Pharos is now machine learning ready, but when you take all this data into account, there's a number of scripts written in R and in Python. So we have parallel differentiate codes, and it sort of assembles a database on the fly based on all this data, which is then given to the metapath encoding system. And in the metapath system, it really tries to evaluate the topology of the associations. In other words, how many different ways it can travel from A to B along a knowledge graph. And once it does that, it essentially runs the extreme gradient boosting classifier, and then it comes up with the predictions, which are then used to impute functions. And we prefer to use negatives that come from experiments. So wherever that is possible, we encourage that, but otherwise by default, we use the OMIM dataset, which is monogenic diseases, to try to associate a set of negatives for the labels. So for example, and probably I should have put this earlier in the slide deck, but this is essentially, once you have that association within a protein and disease, you give that a 1 in the classifier, and if that association is absent, it gets a 0, obviously. And this degree of weighted path counts gets submitted to the classification model. And here are 2 models that we built for Alzheimer's disease, using a set of 53 genes known to be associated with the disease. And this is, and then about 4,000 genes from OMIM that were negative. And between these different models, there is quite a clear difference. So we chose this one to validate. And this is now accepted with revision in communications biology, but essentially we have come up with the top 20 genes classified by our algorithm. We ran multiple layers of validation, which are briefly summarized here. So we looked at pluripotent stem neuronal cells. We looked at the autopsy from folks that had died of Alzheimer's. And then we also did siRNA on a specific cell line that looks at tau filament generation. And out of these 3 genes that are completely novel in association with Alzheimer's, 2 of them really point out to innate immune pathways. And this is the string that shows that that association makes context from a biological perspective. I also thought I was going to switch gear a little bit in the last part of my talk, so the last 3 slides, what I really wanted to show you is that when you have data integrated in Pharos, you can actually start to ask different questions. So one of the questions that intrigued me over a long period of time is, how do we know that a specific chemical ends up in a specific subcellular compartment? So it turns out that when you query data in Pharos, you can get that type of data for about 2100 proteins, where you have small molecule bioactivity data collected from Campbell and PDSP. But you also have, and this is the psychoactive drugs database from Brian Roth. And out of these 44 different compartments, we had to discard 4 because they had fewer than data points. But I apologize for the busyness of this slide, but it's just to illustrate that except for very few that have numbers in the tens, the majority of them include hundreds of or even thousands of chemicals that have been observed to bind to a specific target. So what we do is we combine the information for the target with its subcellular location based on experimental data. So we pretty much make the assumption that at least a chemical has to get in that compartment. And essentially we have built more than 22,000 models using a variety of descriptors and based on scikit-learn and RTKit and FalseServe. And at the end of the day, we have actually 3 hierarchy types models. So we have a larger type of model that looks at plasma membrane, cytoplasm, nucleus, and cell replication. And then for hierarchy 3, there are some that can be observed. And then, of course, hierarchy 4. So, for example, with the cytoskeleton, you really have to go into more detail, or even the endoplasmic reticulum allows you to get that type of variation. So all these models, of course, can be visualized, and you can see that there are multiple families of models that we pretty much we do a decent job with this. We're just about to roll out the publication. So my take-home message is, I think there is tremendous progress in studying the biology of dark targets. However, the caution is that even when you understand the biology of the target, to get from that target to a mode of action target, which is essentially from T-bio to T-clean, it would take 12 to 20 years. So it's not really an easy path. But I believe that predictive models are increasingly more important to assign disease association or phenotype association. And I think I showed you very briefly that in Alzheimer's, for example, you can do that sort of work. And I think for the first time, we can study subcellular ligand location at scale. And with that, I will stop. And thank you for your attention.
Excellent. Thanks. Thanks very much, Tudor. So as I mentioned at the beginning, we're not going to take questions just now, but do type into the Q&A box if you've got any questions for Tudor. We're going to move on now to Melanie Schneider. So Melanie's with me at the EBI. Melanie's going to talk to us about target tractability. So I think nicely complementary to what Tudor has just described.
So hello, and I hope you can see my screen. Thank you for the nice introduction, and I would like to thank also the organizers for the opportunity to present within the Target 2035 webinar series. The topic of my presentation is target tractability, which is a key decision point in drug discovery. So I've subdivided my presentation into different parts, answering the questions, what is open targets first? What do we mean by target tractability? How do we perform tractability assessments? How can you access the assessments? And finally, what are the caveats when integrating different data sources? So, Open Targets is a consortium of academic, and industry partners. So it is a pre-competitive partnership where all partners bring their respective expertise to collaborative projects with the overall goal to systematically identify and prioritize targets. Target identification and selection is the first step in drug development, and therefore very important. The big question at the beginning of a drug discovery campaign is how to identify the most promising targets. And there are two aspects we can investigate. One is the target disease association, asking the question, does target modulation translate to disease modulation? And this is often referred to as target quality also, which you can see here on the y-axis of this graph. The second is concerning the target properties, asking the question, how likely is it that a given target can be used or modulated? And this is referred to as target tractability or ligandability. So in other words, it's the likelihood of identifying a modulator that interacts effectively with the target. Then what we want in the end is a high druggability, but as this is the ability of a protein to bind a drug-like modulator with a therapeutically useful level of affinity, efficacy, and safety, this is very complex and not really applicable unless the project is already very advanced. So this leaves us with target tractability and the target quality. And for the rest of the presentation, I will focus on target tractability. So how do we perform tractability assessments? Here's an overview of the procedure. We have developed a tractability pipeline in a high throughput pipeline. So every target that runs through this pipeline, this tractability file gets assigned to different tractability buckets depending on the rules associated to the different buckets. And then the targets that fall into a certain bucket are put into different, more global tractability categories. The final tractability assessment output is a spreadsheet containing information on a bucket and category assignments per target, which is provided to the user. This information should provide the user with first hints on how to prioritize different targets, but still as a subsequent deep dive on selected targets needs to be performed to obtain a complete and detailed picture of the targets. With our tractability pipeline, we're performing target assessments for different drug modalities, which are the two most common ones, the small molecules and the antibodies. And we're also developing new workflows for new modalities, such as degraders. Now I'm going to explain in detail the underlying workflows for the two most common modalities. So first, the small molecule workflow assessments. Here you can see at the top bucket type, the data source where the data is coming from, the actual assignment rule for each bucket, and here the bucket numbers. For example, for the first three buckets here, the bucket type is clinical and the data is coming from Campbell. For each target, a bucket gets a binary assignment, either 1 or 0. So if a given target has, for example, an approved small molecule drug, bucket 1 gets a 1, and if not, a 0. And if this target has a small molecule in clinical phase 2 or 3, bucket 2 gets a 1, and if not, a 0, and so on for all the buckets and all the targets. As you can see here, we make assessments and use of different data sources that contain different types of information, such as structural ligands, pocket evaluation, and the targets family. Now, comparing with the antibody workflow assessments. So buckets 1 to 3 are highly similar to the small molecule workflow and investigate clinical precedence, as you can see here. The only difference is that instead of small molecules, we're looking for antibodies in our data source Campbell and not for small molecules. Looking at the other buckets, they are very different from the small molecule workflow and are related to the location of the target. As for an antibody approach, the target needs to be located in the cell membrane or be secreted to be accessible for an antibody. Here we use cellular compartment information from different data sources. And depending on the information confidence, the targets get finally assigned also to categories. For the small molecule, those categories are the clinical precedence, and here bucket 4 and 5 group to discovery precedence, and then also predicted tractable. So for the antibody workflow, those are the first clinical precedence, predicted tractable with a high confidence, and then predicted tractable with medium or low confidence. So what happens when we run the whole protein coding genome, which codes for approximately 20,000 proteins, through our tractability pipeline? And here you can see the number of targets that receive a positive assignment in each bucket. Among the small molecules, here on the left side, I would like to highlight buckets 4 and 5. For example, to develop a small molecule drug, it might be highly beneficial to have a target structure with a binding ligand. This is captured in bucket 4 here. With assignments of over 3,000 targets. It might also be good to know if there is information on ligands binding the target. And the data is coming from Campbell, and this is captured here in bucket number 5. In comparison, when looking at the antibody workflow, we see that the number of targets with approved drugs here in the first bucket is much lower compared to the small molecule workflow. But most of the location bucket numbers are generally rather high, meaning that more information is available on target location. This is more apparent when looking at the highest tractability categories per target. Here, each target is assigned to one category depending on the highest filled bucket for this workflow. For the small molecule workflow, 74% of the protein coding genome falls into category unknown, meaning that no bucket could be assigned, whereas only about 49% are unknown for the antibody workflow. So this highlights that there's still a lot of work to be done to shed light on many of those targets. Now coming to the question, how can you access the tractability assessments? The answer is via the Open Targets platform, which you can see here in the center. The Open Targets platform is a data integration and analysis tool for systematic drug target identification and prioritization. It integrates over 20 public data sources to build and score target disease associations. And it supports user workflows starting with the search for either a specific target or disease phenotype. Or you can also use the batch search, providing a list of targets here. The platform is open source and freely available around the world, and the data is available for download or via a REST API. So when you go to the download section on the Open Targets platform, you can find the genome-wide tractability assessment spreadsheet for download. So, but how does it look like on the website? Here I use the batch search with 3 different targets. In the tractability information section, which you can find here, we find the assignments to the global tractability categories, which are here, for the workflows, small molecule, and antibody. When clicking on one target here, for example, we get to the target page. And in the target tractability section, we find information on the bucket assignments here, grouped under the different categories. So this was on how you can access the data. And here's an example of how it has been used. The publication by Behan et al. Is a genome scale application of the prioritization of oncology targets with CRISPR screens and tractability assessment. So they perform genome-wide CRISPR screens in cancer cell lines to assign each gene a target priority score, which you can see here, and then combine it with a target tractability information for pharmaceutical development, which you can see here. So before finishing my presentation, I would like to tell you also about caveats when integrating the different data sources. So there are several points that are important to have in mind. And starting with data extraction, sustainability and scalability, the question is the data easily accessible, for example, via API or download, and upgraded regularly needs to be asked upfront. So it is a big difference if the data is provided through a publication, for example, as a downloadable supplement spreadsheet, which is a one-off event, or if the data is provided through a database or service that is updated. It also makes a difference how large the data is. So if data required from a source is large and the source needs to be accessed many times to retrieve all the data, and additionally the data is updated once or twice a year, as for example for Campbell, one might think to have a local copy of the data, as this enables the fastest access. So on the other hand, if the data is updated on a weekly basis, and this is not really an option to have a local copy, and a performant API is required. This is, for example, the case for the PDBE. Not only the access, also the processing needs to be fast enough to provide sufficient scalability. So this often restricting the type of analysis we can perform in a high throughput mode, as we want to do with this pipeline. So when looking at the actual data itself, we need to be aware of data heterogeneity, different data standards that can be used, and data quality issues. So ideally, one should know where the data is coming from and which standards are used and how it has been collected. But depending on different experimental setups or samples and other conditions, the data might be so heterogeneous that comparison between different targets is not directly possible. Luckily, some of the data sources, such as Campbell, already tried to correct for those issues, but this is not always possible. Finally, I would also like to highlight the importance of data presentation. in particular to avoid giving an unwanted or false impression and consider the diversity of the users. So depending on the user's expertise, one might prioritize and interpret data differently. Also, depending on the actual project at hand, someone looking for a drug repurposing opportunity might use the data very differently than someone looking for a new target. So those are also the main reasons why we prefer to provide tractability assessments in an, as we think, rather objective way, without too much of a judgment, or to finally give the user enough flexibility to adjust the evaluation to the specific case at hand. So with this, I'm coming to an end, and I would like to thank all the partners that contributed to this work. And I would like to mention that I'm always open for new ideas on tractability assessments, so feel free to contact me. And I will also participate in the Q&A session later. Thank you.
Very good. Thanks very much, Melanie. Yep, so I can see some questions coming to the Q&A. So please all continue to type those. And I'm going to hand over now to Brian Scheukert from UCSF, who is going to talk about ultra large scale virtual screening. Melanie, I think you might need to unshare your screen. Okay, great. Thanks, Brian.
Okay, thanks, Andrew. Thanks, everybody. So I'll just, time is short, so I'll jump right into it. What's changed our world in structure-based ligand discovery recently is the explosion of available molecules, and that's really happened primarily through the efforts of Yuri Moroz and his team at Enamine, and on our side, John Irwin and his efforts to turn the Enamine make-on-demand library into, small molecule structures that can be docked. And I'll just say quickly that the foundation of it is a huge investment on the part of Enamine on building blocks, often stereochemically rich building blocks, interesting chemotypes, put together through one of about 140 2 and occasionally 3 component reactions that they have under good experimental control at Enamine. And that results in really functionally rich molecules, often congested functionally, a lot of stereochemistry and 3-dimensionality, really often neat looking molecules. Among the bond forming reactions for those interested in this sort of thing there's I think about 11 carbon-carbon bond forming reactions. So they actually access a pretty interesting range of reactions and chemotypes. The size of the library now exceeds 14 billion molecules. On our side, through John's work, about 1 billion of them have structures calculated. So we're sort of lagging the enumeration of different molecules. We were at the beginning, we were pretty skeptical about this enterprise because we worried that molecules could not be accessed, so they could be enumerated and they could be docked, but you couldn't get them. We worried about that, and then we worried that maybe they weren't so interesting. But we, we're soon convinced that A, they were in fact quite interesting, and B, we could actually get our hands on them for pretty quickly. And so the challenge became, well, can we actually make something out of this huge new space through structure-based docking or other technologies? So our first proof of concept studies, which have been published, so I'm just going to go touch on them because they illustrate some points, were against beta-lactamase, which I'm not going to discuss, and against the D4 receptor in collaboration with Brian Roth's lab. And there we docked this the size of the library that we had then, 138 million molecules, each one at a time in multiple orientations and configurations. So, pushing, trillions of complexes. The calculation, setting up the calculation was a lot of work. It usually is, and analyzing the calculation was a lot of work. But actually prosecuting the calculation once everything was ready went pretty quickly. About 2 days on a decent sized academic cluster. One of the things that seemed ambitious at the time, still seems ambitious, was usually when we run these docking screens, we'll test 30 to 50 molecules, and that was all. Even when we were docking 3 million molecules, which was the size of the space available to us before the advent of these ultra large libraries, testing 30 or 50 molecules was probably silly, but it seemed really silly now that we were docking hundreds of millions. So in this experiment, we decided, okay, we're going to test a lot more compounds, which meant synthesizing. Each one of them had to be synthesized de novo. They were all, as far as we know, new to the planet. So it was a big enterprise, but we thought, okay, let's not just test the high scoring molecules like we usually do, but let's test molecules that across the docking scoring range. So we get some idea of how doc score predicted experimental results. And I'm going to get to that in a second, but we were, of course, also interested. We had to keep the interested of our patient collaborators in the Roth lab. We were also interested in getting molecules that were potent and selective for the D4 dopamine receptor, it's a G protein coupled receptor, and we were able to. So this is this is the best performing molecule. It's straight out of the dock. It has an EC50 of 180 picomolar, so it's it's quite potent and super selective. It doesn't detectably modulate the D2 or the D3 dopamine receptors, which are the closest to the D4. So had some other interesting properties. I just want to point out that it's sort of characteristic of the sorts of molecules you get out of this enterprise. It's highly stereogenic, really three-dimensional. It's got 3 stereo centers. 2 of which we had to purify. So we had to ultimately purify 4 diastereomers. So some interesting chemistry comes out of this. And there were other ones. So this was the best, but the second best was a 10 nanomolar pure agonist, and the third best was an 11 nanomolar mixed agonist, and the fourth was 20, and so forth. So we got some pretty potent molecules out of this. So getting to this idea of how does the docking predict the experimental results? Well, in terms of affinities and even rank ordering, we don't expect it to, but we got this pretty interesting result where as the docking, so the docking scores on the x-axis and the hit rate, okay, so the simplest evaluation of success, how hit rate is the number of molecules that are active divided by the number that are tested. And here we define active as 10 micromolar or better, right? And you can see that there's this sort of looks like a well-behaved curve where at the highest, the best, the most negative, so the best docking scores, the hit rate is pretty high, about 24-25%. And as the docking scores decline, so too does the hit rate, and this sort of looks like a dose response curve, right? And until you get to these low docking scores where the hit rate falls to 0, and as you go lower and lower, it stays at 0. It doesn't bump up or anything. And the numbers here on the curve are the number of molecules tested in each scoring range. So we think we had enough molecules tested to make this sort of curve reliable. And I got to say, this was all done in Brian Roth's. All the experiments were done in Brian Roth's lab. So 549 molecules tested, I think over 80 80, the ones that were active were tested in full dose response by sort of a heroic postdoc Sheng Wang, who now has his own lab in China. So there were a couple of really interesting things that came out of this. One of them was, because we have scores for all the molecules that were docked, and we have a model for how score relates to hit rate, we could predict how many actives there were in the library at 10 micromolar. And it turns out to be an astonishing number, 452,000 molecules predicted to be active at the D4 receptor, which is far more than anybody can test, far less synthesize. But the other sort of disturbing thing that comes out is that this curve, if it looks beautiful, but it's flat at the top. And, we're sort of trained to think that that's the way curves should look. But actually what this teaches is that the docking score isn't informing us in any way about hit rate when you get to the very top of the curve, which is the part of the curve that you're most interested in. It's flat, and this is something we're struggling with, and I'll come back to. Okay, so just very briefly to say that we've really invested in this technology subsequently, and have been using it to discover and progress compounds in a bunch of different targets. And this is the message here is just that this wasn't a one-off. We've had that success, and so have others, with this overall ultra-large library structure-based approach against multiple targets. And there is a range, and it doesn't always work. We've also had some disappointing failures, and if not failures, places where we don't get very potent molecules, but certainly some targets we have. And I'll just focus on one of them, the sigma-2 receptor, because it gets back to illustrating some of the challenges and opportunities. So this is a collaboration with Andy Cruz's lab at Harvard Med School. And we're going after this receptor, the sigma-2 receptor. I think by Melanie's categorization scheme, you'd call it tractable. There's a lot of molecules that are known to promiscuously bind to the sigma-2 receptor. It's sort of a well-known dark horse in pharmacology. It's also an orphan. Exactly what it does is really still not clear. And there's few, if any, selectable molecules for it to select for it, for instance, versus the sigma-1 receptor. And so that's something we were definitely interested in looking for. So this was also, this campaign was also run by this talented postdoc in the lab, J.K. Liu, as was the D4 campaign. And so we did the usual docking thing. We took every molecule in the library and docked it against sigma-2. So there's, by now, 500 million in this campaign. And it took about 2 days. Here we tested initially, we've tested 79, but as you see, we're gonna test another 500 again. But so far we've just tested the top scoring molecules that came out of the screen. We haven't tested the low scoring ones like we did against the D4, but that is our intention. And one of the things that we want to get to in this campaign is, can we, so that this beautiful dose response curve, docking score versus hit rate, has only been done once. And, the one time it was done, we have this issue of this flat upper baseline. And so we wanted to see if it was even remotely reproducible. And two, can we, we've been improving, we think improving the docking scoring function, and with these improvements, can we start to see a sloped upper baseline? So we start to get information out of that upper baseline. Anyway, so here's the still preliminary results. They're preliminary. The data are all very good. They're all full dose response curves. So we're quite confident about the data, but we haven't tested, we've only tested the top scoring molecules. But anyways, here's the results so far. So of the 79 all high scoring molecules, 52 were active, and here active means at 10 micromolar. So, a 66% hit rate. It's, don't try this at home sort of thing. This target is really well suited to binding small molecules. So it's, in that sense, 66% is better than we expect to get normally, far better. But anyways, let me just show you a few of them. This molecule is a 34 nanomolar Ki on the sigma-2 receptor with decent selectivity versus sigma-1. This guy's 22 nanomolar. And so we get, just to say, so we get some potent molecules with diverse chemotypes. All of the molecules chosen were from different chemotype clusters, so they all look different. I'm showing you the ones that are selective. We don't right now, among our problems is we don't predict selectivity very well. They weren't counter screened versus sigma 1. So the selectivity is just a function of the number of molecules we're accessing and the novelty of the chemotypes. That's what we would say. Just to show you that, Asaf Alon in Andy's lab, a fabulous postdoc, has managed to solve a few structures of these novel chemotypes against the sigma 2 receptor, and I'm just showing you two of them. This is one of the molecules, and the yellow structure is the docking prediction, and the blue and green just from two monomers in the asymmetric unit are the crystallographic results. So they superpose just as accurately as we could hope to them, given all the inaccuracies in our method. And this is the second molecule. Same thing, the yellow is the docking prediction, and the blue is the crystallographic results. And once again, it's, there are a few differences, but basically it's right for the right reason. So that's very comforting. I know I'm probably running out of time. So this is, I think, where I'll end. So, we don't have the full answer here, in terms of can we, do we regenerate something like this sort of docking response curve? We've only tested molecules in the top scoring region. We have selected and ordered and actually had the compounds delivered molecules that are across that docking scoring range, but we don't know the results yet. That's coming. But we're getting hints that maybe we will see some tilting at least in the upper baseline. So this is the docking, it says energy, it's the docking score. It doesn't mean anything except for the sigma 2 receptor. But these are how the docking score goes. So more negative is better, and percent inhibition is on the y-axis. So a lower value is better. So you can see that at the very highest energy tranche, where there's 9 compounds were tested, 8 out of the 9 inhibit at 1 micromolar bounds to sigma 2 receptor at 1 micromolar. And then in the next lowest energy, the next second best energy bin, 12 out of 17 bounds, so 70%. And then the third best energy bin, about 60%. So you can see that as the energy falls, or as the docking score gets worse, that's a better way of saying it, the hit rate declines. Now, whether that's going to be true over the curve, whether we generate a curve at all, I don't know. But that's something we're hoping to see, and seeing if the new methods that we've introduced, which were actually just optimization, have improved things. I guess so. This is the last thing. I'll end with this. I think the thing for the take home, the big take home for the community, is that accessible chemical space is exploding. These molecules are available to anybody. We describe them on, John Irwin describes them on the Zinc database, but they're available directly from Enamine and from Wuxi and from a few other suppliers. And so we expect that from an dockable standpoint, about 10 billion will be available and will be using for sure in the next few years, and over 1 billion are available right now. So there's all sorts of problems with that, huge challenges, but I think it's a super exciting time for the community. Thanks very much.
Great, thanks, Brian. Excellent, 3 great talks. We are just seeing the Q&A sort of gaining some extra momentum now. So we've got sort of 5 minutes of sort of verbal Q&A. I'm going to try and just pull out 1 or 2 of the questions. I know, Brian, you haven't yet had a chance to look at the ones that are coming through, so I'll leave the more detailed ones for you, but maybe just a quick follow on to what you've just talked about. There's the question from Pankaj asking about whether the docking score versus hit rate curve could be piecewise linear, kind of a threshold function rather than sort of a dose response curve, and whether there are any implications of that. I don't know if you have any thoughts on that question that you have now, or whether you want to think about it and type an answer in.
Well, yeah, I should probably think about it, but my first instinct is to say that, the whole dose response is like, for sure it's not that, right? There's no reason for it to look that way. But It just sort of does, and for now. So I don't expect that to hold up in that functional form, right? So a threshold function is probably a better way of thinking about it.
Great, yeah, makes sense. Okay, thanks. I'll let you kind of take a look at what people have been typing in. There's some specific ones about D4. Melanie, hopefully you're there. Just saw a question from Josh Pottle asking about the multiple functions of certain targets or drugs, mode of action, agonism, antagonism, allosteric modulation, and so on.
So the question is exactly, I think I didn't get through all the questions in the Q&A. So
No. Is there I mean, that's fine. I was just going to ask, I'm just asking this one verbally just to see if
yes,
there's a quick answer. So this is there's a question from Josh Pottle asking about whether Open Targets considers, I guess, mode of action for certain targets or drugs. So agonism, agonists or antagonists, allosteric modulators, you know, that sort of information.
I see. So mode of action is included in the target disease relation. So there is some information which is not part of the tractability assessments, but yeah, it's also available on Open Targets. But I think it's not as detailed as agonism or antagonism, and because I think for every protein or every receptor, this might be rather a gradual transition between the two, because there are also partial agonists and partial antagonists and so on. So I think they don't go so deep into the details.
Okay, great, thanks. Tudor, I know you've answered a couple of rather detailed questions online, which I'm hoping everybody can see. But maybe just a question from me on the Alzheimer's work that you described. I mean, you sort of hinted at there being certain types of data that you believe are sort of reinforcing the relevance of those targets in that particular disease indication. I just wondered if there's any more you could say about the whether certain data types or factors contribute more in those models that you're building, or whether you can unpick that and sort of understand, you know, certain diseases, you know, this is what really matters. Other diseases, you know, this other type of information is more important than certain other things, or whether it's just a kind of a black box and it's difficult to unentangle.
So that's a really good question, Andrew. What we've played with and observed, there are differences. So for example, in the input for machine learning, by default, we use the links dataset, CCLE, the cancer encyclopedia expression data, etc., etc. And then it turns out that in reality, what you really want to do is test a multiplicity of models where you deliberately exclude links and CCLE and whatnot. So what we found, for example, for Alzheimer's is that you get a better resolution and more informative models if you exclude all this cancer expression data, because it's really not based on neuronal phenomena, etc. So yeah, you're spot on with that observation. I just didn't have time to go into details about all these things, but we currently run multiplicity of models with a variety of descriptors, and yeah, so that would be a much longer conversation.
No, thanks. That's really interesting too. That's a case of maybe more is less.
Yes, so in this case, we found, for instance, expression data and protein-protein interaction data to play a more important role.
Great, excellent. Okay, well, thanks very much, you three, for getting us off to such a good start. I'm going to try and keep the time, and so we're going to move to the second set of talks now. The first of which is questions, please, to Wendy in the Q&A. I can see one already, but I'm sure there'll be others as well. And so we're going to move on now to John Corso, who is from ZAI. And John is going to talk to us about probe discovery for undrugged targets, continuing with the AI theme. So John, all yours.
Great. Let me just share my screen here. Is that coming through?
Yeah, very good.
Great. Okay, so thanks, Andrew. As you heard, my name is John Quazzo, and I head up drug discovery here at ZebiAI Therapeutics. Just for your information, ZebiAI is a brand new company. We're located in Waltham, Massachusetts. We got up and running about 6 months ago, and we're really looking forward to today when we can all be together under the same roof. So we're a pandemic-born company. But today I'm going to tell you about an exciting new machine learning-based approach to identify small molecule probes for undrugged targets without the need for protein structural information. So before I go into that, I just wanted to review some of the goals and challenges of the Target 2035 initiative to unlock druggable biology. So the goal is lofty. It's to generate a complete set of pharmacological modulators for most of the 20,000 or so human protein coding genes in the next 14 years. And only a very small fraction of human proteins right now, anywhere, you've heard the numbers related earlier today, have the minimum quality chemical probes that meet, the key potency, cell activity, and selectivity criteria that you need in order to begin to understand the biology of a particular target. And also, a good chunk of protein targets don't have protein structures, PDB structures available as of yet, although the gap is rapidly closing there, which is exciting. However, this is really a perfect opportunity for the technology I'm going to describe to you today. And our top technology leverages breakthroughs that we've developed to screen thousands of proteins and create machine learning models using automated methods. And this is done in partnership with Google Accelerated Sciences and XChem. The XChem that's located here in Waltham, Mass, just to differentiate from the Oxford XChem group, who we've recently been in contact with. The speed and economics of this new approach that we call the Genome Initiative really opens up the possibility of identifying these potent cell active and selective small molecule probes within the Target 2035 timeline, and I'll walk you through that today. So here's an overview of our structure-independent approach to machine learning model and probe generation. We began with a target protein that's supplied by an external researcher. The SGC and EUV Open teams have really been fantastic partners here, supplying us with really, really high quality, high purity, well-behaved target proteins. We run a DNA encoded library screen with 10 to the 11th diverse compounds, so 100 billion. Many times we run this screen under multiple conditions. So we run target by itself, we run a no target control, we run target plus a known competitor if we have that available to us. We can also run off target side by side, targets you want to avoid if you hit your primary target. And we process the millions of resulting data points that come out of those screens. This data is then used to train machine learning models. So we do it in pretty standard fashion where we withhold part of the data to validate the model, and that once we see we can predict the withheld data and have a validated model, we use the model to select readily available and very reasonably priced commercial compounds from the large virtual libraries that you've already heard quite a bit about today. These predicted compounds are sent back to the collaborator for testing in their own hands. Once they've identified active chemotypes, we can run another machine learning cycle back at ZebiAI to refine the structure-activity relationships and focus in on desired properties for the disease area of interest. We've recently shown that we can run targets expressed in cell lysates without further purification, which greatly accelerates the process. So overall, it takes about several months. And one of the most appealing aspects to me is that we do this as an open science initiative. So the Genome Initiative is open science, and the researcher commits to publish their results in the probe. So finally, the researcher has an option to pursue a drug development program with us and fully engage our biology, medchem, computational chemistry, and data science teams. So let's move on to some examples. So the first example I'm going to show you is a pretty well-established drug target, estrogen receptor alpha. with a significant amount of chemical equity. We ran this target as one of our initial POC experiments with the Google team. We first generated 2 machine learning models, as you can see here on the right, with the Dell screening data, a graph convolution-based neural network, and a random forest model. The scientists who produced these models were actually blinded to the target, so they couldn't use that information to bias their approach. The models were then used to predict binders from both, in this case, we used MQL and an internal virtual library. Just over 100 compounds were selected from each of these sources and tested against CR alpha using a competitive binding assay with a fluorescently labeled probe. And the results were really exciting. We saw hit rates of between 20 and 35% for the predicted compounds, with the GCNN here outperforming the RF by a bit. Potency ranged from 30 micromolar in the lighter color down to as potent as 10 nanomolar in the darker colors. Even more exciting to us was that these models were predicting compounds in both known and unencumbered IP space, as well as binders that were pretty dissimilar to the nearest neighbors in the DNA encoded library dataset. So this really showed us for the first time that these machine learning models trained on this data can understand the preferences of a protein binding site, really get a sense of the shape of that site using the DNA encoded output library data alone. So all of this work, along with similar results for 2 other targets, are published in this past summer in the Journal of Medicinal Chemistry, McCloskey and Siegel. So please take a look at that paper for further details. So we also performed another round of optimization on the ER alpha compounds by running similarity searches on the 6 different chemotypes that we initially had identified. So compounds from these similarity clouds were run back through the machine learning model, prioritized by their scores and assayed. And on the y-axis here, we indicate the fold increase in potency. So all series, once we built these similarity clouds, reran and tested these molecules, showed a significant improvement in potency, as highlighted here by this green ellipse. We saw, in most cases, we saw improvement of 3-fold. In a few cases, we saw 20 to 40-fold improvement. We also began to see correlations between the prediction scores and the increased potency. And again, we didn't use any crystal structure or protein structure information as part of this optimization process. So in this work, in this experiment, we saw that not only identify, but also improve the potency of the molecules predicted by the ML algorithms. One of the critical properties of a probe is cell permeability, as I mentioned earlier. So we tested our machine learning compounds in both agonist and antagonist mode ER alpha cell-based assays. The agonist assay titrations are indicated here in red, antagonist in blue. We see that our compounds are clearly cell permeable and showed several different mechanisms of action. We see antagonists, we see a mixed response, and pure agonists.
Hey, John, this is Tudor. Your slides aren't progressing anymore on the screen.
Oh, really?
We see novel multiple AR alpha actives identified. That's where we are. Sorry, didn't mean to interrupt.
Tudor.
Andrew, are you seeing it?
No, we're still seeing the novel, multiple novel AR. Slide 4 we're on.
Okay, let me stop sharing and then see if that works. How about that?
It's just coming up.
Is that coming through?
No, we just have a blank screen, I'm afraid, at the moment. It does say that we're seeing your screen, but I'm not seeing anything. Do you want to have another go? Maybe sometimes it just needs to.
Okay, stop the share one more time. Sorry about that. Screen share. Let me just do this 1 second. Let me just take this. Hang on. Just escape out of presentation mode for a second. See if that works. How about now?
Yeah, there we go. So we can now see slide 6.
Okay. How about if I go into presentation mode? Is that, you know?
I mean, it's good as it is. I mean, it's very clear as it is. If that, I know sometimes presentation mode causes problems. So if you want to stick with this mode, John, that'll work, I think, pretty well
Okay,
for us.
great. So we're on slide 6. You see that?
Yeah, all good.
Okay.
Right.
These are the traces I was referring to. Agonist, antagonist. So it made sense that we saw all these different modes of action because we had identified these compounds only based on their binding. We hadn't biased for one particular mechanism of action over another, so we were able to collect all of them. What was also great was that these targets showed on-target activity because we did not see any activity when tested in an estrogen-independent cell line. So just from these results, we see that the machine learning derived compounds are cell permeable and show on-target activity in cell-based assays. So I'll advance here. There we go. So not all targets can be extracted and purified from cells. So we also ran a selection with ER alpha produced in a mammalian cell lysate. We trained a model on the resulting data set and saw a hit rate of about 50%, which was exciting. Number of advantages of screening in lysates. First, it's a very rapid production and isolation of protein in its native environment with all of the normal post-translational modifications and PPI partners. And then using lysates may enable our machine learning approach for very challenging targets and potentially even target complexes. So these results show that successful machine learning models can be built from partially purified protein data, and really strongly suggesting that the models can identify patterns even in noisier outputs. So far everything I've gone through is for ER alpha, which is, as everybody knows, a well-established drug target. But what about previously undrugged or even unliganded targets? So our collaborators at the SGC have been great, feeding us a steady stream of very challenging targets with no known small molecule activity that have been worked on for quite some time. One such target is the E3 ligase, DCAF1. We're excited when the Toronto team shared their initial SPR results with us that showed that our molecules had bound to the target. We're even more excited when Levon here let us know that he had solved co-crystal structure at a resolution of 2.3 angstroms with one of our molecules. Hopefully we'll be able to release the structure at some point in the near future. Apologies for blocking it out here. The molecule itself is small. It's less than 400 molecular weight with excellent properties. And there's obvious opportunities for future optimization, further optimization, which are currently underway. Using, taking advantage of the co-crystal structure. What's even really more interesting is that the molecule is oriented in a way that's optimal for the development of potentially new DCAF1 based degraders. So these results demonstrated we could identify interesting starting points for an undrugged target. We did have an advantage with this target, one advantage in our screening campaign. We were able to use a peptide ligand supplied to us by the SGC to define competitive and non-competitive DEL binders. And that helped the machine learning model focus on the chemotype that we identified here. Forward. Oops, I want to edit. Sorry. There we go. So selectivity obviously is also a critical property of probes. WDR12 is another undrugged target supplied to us from SGC Toronto. Target was even more challenging than DCAF1 because we didn't even have a competitor peptide to use in the selection. So we simply did target and no target side by side. Our machine learning approach produced multiple chemotypes that bound this target, as the SGC verified again by SPR results. We also ran two other targets, two other WD targets, WDR5 and WDR41, side by side with this one, and observed that the WDR12 compounds, as seen here along the bottom, are not predicted to bind to WDR5 when we ran them through the WDR5 GCNN model. So here's the score of WDR12, high as you would expect in the WDR12 model, but when we ran them in the WDR5 model, we got barely observable scores there. They don't even show up on this chart. It was great to get the results again from the SGC because they ran SPR against WDR5 and showed that the WDR12 binders do not bind to WDR5 as had been predicted by the machine learning model results. So in this case, we've shown that we can identify initial starting points for probes for a really challenging target class that also show selectivity to another protein with a structurally relevant, related domain. So just to finish up here. So what I've hoped that I've been able to show you today is that our new machine learning platform is optimal for rapid probe identification, structure independent machine learning approach that can rapidly generate predictive models for multiple target classes in parallel. ZBI machine learning models can identify novel chemical starting points from the vast virtual chemical space in both probe and drug discovery. I haven't talked about our drug discovery efforts today. We have a considerable amount of work and success on that path as well. Machine learning predictive models show on-target activity in cells. The models can provide information on compound selectivity required for effective probes. And our models can also be built using data from purified or lysate-expressed protein targets. The models can be used to optimize binders for both potency and selectivity, and we can also supplement them with assay data to iteratively improve the model throughout a program. I haven't shown data on that. Other data sources can obviously also be incorporated into our machine learning approach, outputs of other screening technologies, structural information, and PK/ADME tox-related data or filters. So just a lot of people involved in this effort. This is a very, very small number. It's a small fraction of the team at ZBAI Therapeutics. We have our data science team, our protein sciences team led by Moritz. Christelle Huguet, our SAB lead. We recently brought on board Rafa Gomez Bombarelli from MIT as our chief learning officer, Rick Wagner, our CEO, Brant Binder, our president. And then the fantastic contributions of the team at SGC Toronto, led by Aled Edwards, and the main connection there is through Peter Brown, Masoud Badadi's team, and Levon, I highlighted his work. Masoud has been running all the assays. XChem team that was involved in all the initial ER alpha work, led by Matt Clark. And obviously, last but not least, the Google Gas team, led by Patrick Riley and Stephen Kearns. So thanks a lot.
Great, excellent. Thanks so much, John. So let's now move on to our final speaker of the day. That's Kate Stafford from Atomwise. And Kate is going to talk to us again on a similar theme around virtual screening, machine learning. So Kate, over to you.
All right, thank you very much. Can you see my screen?
Yes, we can. I can. Good. Take it away.
Okay. Thank you for inviting me to speak, and to the audience, thank you for sticking with us till the end of and the last talk here. So my name is Kate Stafford. I'm a senior scientist at Atomwise. We are a company based in San Francisco. We're about 60 people now. So I'm coming to you from the cheminformatics team there. And I'm going to talk a little bit about, I guess it's sort of fitting that this talk is scheduled at the end, because I wanted to talk about what happens after you have performed your virtual screen and now you want to optimize your lead and move on beyond your initial screening hit. So let me go ahead and first sort of set the stage with a short summary of some of our successes as far as virtual screening projects goes. This is a summary of our academic collaboration program, which is called AIMS. So these numbers are actually a little bit out of date. These come from this past summer. So we've improved on our total number of projects so far. Since this data set was compiled, the hit rate is still about the same. So we're looking at a very diverse range of academic projects that have been brought to us by our collaborators. A large number of targets, challenging targets, many cases where we needed to build a homology model, where we did not have training data available, or where we had some sort of unusual challenge around the binding site itself, such as looking at a protein-protein interaction. And from any audience that includes Brian, I don't have to sell you on the importance of ultra-large libraries. This is also something where we have been investing significant effort into in both the algorithmic side and on the infrastructure side, is how we can bring some of these ultra-large libraries to our partners and be able to use them at scale. So we've developed this sort of hybrid methodology that approaches, that begins with a sort of ligand-based approach and saves our expensive deep learning models for those areas of chemical space that are most likely to be productive and interesting and for further exploration. So I want to start by asking what exactly, how exactly do we build on the success of this sort of virtual screening applications? A point that Brian made in his talk earlier is that the more that we screen these very large libraries and the more diverse and interesting chemical space that we have access to, the more opportunities we have for success with more interesting compounds with higher initial affinity out of the screen. And the more virtual screening performance, the better we get at that task. The more hits we have, and the higher the demand becomes for follow-up work on hit to lead, lead optimization, selectivity optimization, as well as questions about bioavailability, ADME, toxicity, et cetera, that I'm not going to talk about today. So what we need to do is think about methods that will improve performance on both types of screening tasks, virtual screening as well as optimization, and those can be quite different actually. They have very different background distributions. Of course, virtual screening is looking for that one positive needles in large libraries of mostly negatives. And the further you get down the optimization pathway, the more you're looking at a data set that primarily consists of positive compounds, possibly not of the affinity that you want, but that's where it becomes much more important that you can actually understand what the model is doing, what it's saying, what features of the particular compound or target are leading to a particular prediction. And you care a lot more about being able to successfully rank order compounds as well as make absolute predictions about binding affinity. We also want to improve performance on these additional tasks like selectivity, counter-screening, anti-targets, and give chemists tools to understand what the model is thinking when it makes a particular prediction. So I wanted to share two simple methods that we've been looking at that are shareable, generalizable beyond our platform, and fit in a 15-minute time frame. So I wanted to start out just by sort of setting the stage on the methodology. If you've been to a AI and drug discovery talk, you've seen a version of this slide before. So this is a brief sort of graphical illustration of grid-based three-dimensional convolutional neural networks. These are sort of the initial methods that were used, stick with the grids for now, that were very directly generalizable and very straightforwardly generalizable from a set of methods that were developed for two-dimensional images. So just by analogy to two-dimensional pixels, you can take your atoms and three-dimensional structures and assign them to three-dimensional voxels in a similar way to the way that you see edges and textures in local features of image recognition. You have biochemical features like hydrogen bonds, hydrophobic interactions, et cetera, that compositionally add up to a prediction of binding affinity. And of course, these little illustrations are what fits on a slide, but these are getting to be, those pictures of humans are getting to be a little out of date. Of course, the image recognition field has dramatically improved over the last several years, and the quality of the predictions that come from 3D CNNs in an atomic and a binding context have also improved. So this is also an area where we are not the only player in town. There are many implementations now available of this particular methodology. I want to recognize the Coase Lab in particular as having been particularly effective in exploring this area. Now, the one limitation of the grid-based methods limitation or opportunity, depending on your perspective, is that they have some challenges around rotational and translational invariance. You can argue that what you get there is that you get the data augmentation for free, and that you have to do some random rotations and translations of each example that you look at in order to make sure that you teach the model something generalizable about those physical features. Now, in the last few years, there's been a lot of interest in graph convolutions as an alternative to, as an alternative model architecture to the 3D CNN. In some ways, this is a bit more of a natural way to think about things, because of where a lot of people with a cheminformatics background are used to thinking of compounds in terms of graphs. So this is an implementation that was introduced by us at ACS last year, which is under the name of AtomNet Graphite. So in our implementation, this actually consumes the same data as our grid-based method, and the same data in this case will be the atom XYZ positions and an atom type. So there's been a lot of interest in the relative qualities of these two types of models. I want to go ahead and show you a boring table of very similar numbers, but that's exactly the point, that both of these have very similar baseline performance on the 6-fold cross-validation ensemble of internally curated but public data, all related to inhibitors that bind the ATP sites of kinases. So there's one difference between those two things, though. They give you very similar overall performance, but they see the chemical world differently, and they make different types of mistakes. There are some examples that are persistently hard for both models, and what's one thing that's actually encouraging about the qualities of these models is that you can actually use those persistently hard examples. Those are enriched in data errors, misannotations, database problems, experimental errors, or unfiltered out of distribution features like covalent compounds that we were accidentally treating as non-covalent. On the other hand, there can be architecture and model dependent biases, functional group biases, target types, etc. Can often be difficult to tell what led to a particular pair being poorly predicted, which means it's also difficult to tell prospectively. So there's an idea that's out there about training these two models, these two model architectures together in a fusion architecture. We thought that since these guys make largely orthogonal errors of largely similar magnitudes, what happens if can you improve overall predictive performance by just training them independently and treating them as an independent ensemble of models? And that's why I showed you that boring table of numbers before. Treating these as an ensemble with the same number of ensemble members in each column leads to consistently better performance than simply taking an ensemble of different models of each one of the other type of architecture. So if you mix architectures, you are always going to be able to see a better aggregate performance in a large data set than if you average an ensemble of models that is only of one architecture. And this is actually effective, small but consistent improvements over a variety of benchmark tasks that works for virtual screening and activity classification. It works for direct PKI prediction. It works for a pose quality classification task, which is very similar to what Wendy Cornell had mentioned in her talk earlier, where there's a pure geometric problem where we're only interested in making a prediction about the correct binding mode, as opposed to predicting the activity of a compound. And it also works in a selectivity context where we're kind of turning the problem around and asking, instead of for a given target, can we rank compounds? Instead asking for a given compound, can we rank the targets in our data set by the likelihood that this compound binds that target? So if you're interested in this question of what the correct model architecture is, which one should you choose for a particular problem, I think the answer is in this meme. Why not both? Always, whenever you have the opportunity, try doing both. And as I said, in our implementation that is able to consume the same data set and be trained on the same underlying data, you consistently see improvements by averaging ensembles. So I want to make I know we're running short on time here, so I'm going to go fast here. Briefly go into a question about model explainability, which I mentioned in the beginning is something that is of interest to chemists who are doing further development in lead optimization, selectivity, et cetera. You want to be able to look inside the black box of these methods to understand the model's predictions. Grid-based methods have sort of an intuitive physical meaning in three-dimensional space. They are complicated, again, by that need for data augmentation, as well as by some sensitivity to the parameters of the model. Graph-based methods, you can kind of interpret them atom by atom, which is potentially more useful in the context specifically of optimization settings. So you can extract, this is pretty straightforward, extract the edge activations from our implementation of that graph-based network, which is all based in PyTorch. You can accumulate the activations over those the filters of the model onto your, just write them out as in the B factors of your PDB. This is an example from, thanks to Henry Van Bedham for providing this example of just an arbitrary ligand in a receptor context. Now, if you look inside the black box at specific examples, these are a pair of compounds that differ by about 2 pKi units. And you can see and map onto the features of these ligands that you actually see activations related to specific features of the three-dimensional structure of these compounds that can be interpretable and the context of their binding activity. You can actually also do this with selectivity. Sterosporine is of course, very broad spectrum kinase inhibitor, and you see very little difference between two randomly selected kinases from the data set. As they interact with sterosporine, they have very little differences in their activations. By contrast, if you look at the same compound bound to a wild type and mutant EGFR, where it is known to have both a different binding mode or slightly different binding mode and a different potency and different binding affinity and activation to these two different receptors. One thing that's very gratifying to see here is that you do actually see the mutation stand out in the visualization in this tool. And just to be clear, the model that produced these evaluations did not have either the wild type or the mutant EGFR in its training set. So it was able to pick up on those features without having seen that data. So I'd like to thank everybody again and wrap up, and I'd be happy to take any questions in the Q&A at the end. Thank you, everybody.
Excellent, great. Thanks very much, Kate. Thanks everybody for the talks in the last session. So we're doing well on the Q&A. All the questions have been answered so far. So we have like 5 minutes left though. So I'm just going to maybe expand, Kate, on what you just talked about there in terms of interpretability, and ask John and Wendy and perhaps also other speakers may wish to chip in as well on this, what their view is on this question of interpretable models. I mean, in some ways it's a philosophical question, and it kind of depends who you're working with and how important it is. But John, Wendy, do you have any thoughts on this topic more broadly? Is it something you're bringing into your models or looking to bring in?
Wait for Wendy. For me, interpretability is, I'm a biochemist, so I want to see, active molecules being produced and generated and improving in their features. So that's that's where the rubber meets the road for me.
Yeah, this is Wendy. Interpretability is obviously really important because that lets you then decide what action to take next. What do you change? It's non-trivial. I think you can get out some initial interpretate or explanations, flagging certain atoms that make sense, but then you really have to start looking at in a more localized way, thinking chemically. And also when you think of dominant, different functional groups and contributions, dominant positives and dominant negatives and vice versa, it gets a little complicated, but certainly you can extract some useful information from these models.
Okay, great. Thanks very much. Maybe one other question, again, sort of following on from what Kate talked about. I mean, you've all described, using some of the latest deep learning neural network and similar models. And Kate, you talked about sort of integrating different ways of doing that, combining models into a consensus. Have you thought about maybe some of the other more, I'm going to say old-fashioned, that's the wrong word to use, but some of the more traditional approaches? When obviously we heard from Brian about, where he's at with, building upon many years of experience in docking, scoring, actively using these methods for real. I mean, is that something that you're looking at as well to yet further sort of advance the predictability of and performance of the methods you're working on at the moment?
For us, definitely. Yeah, we have a computational chemistry team, and we've been, actively, mixing those efforts with our DNA-encoded machine learning outputs to further our drug discovery programs. And it's been really exciting. The results have been great.
Yeah, we're definitely absolutely interested in having good models. We talk about three-dimensional structures and being a structure-based drug discovery company, and that means that we really do care about having good models of poses, how the compound interacts with a receptor, and that means you got to do some docking. We do some docking. We do some pose sampling around our complexes of interest. We have a lot of interest in that problem that I think Wendy and I both touched on, is just ranking poses, ranking models of particular binding drug target interactions, and making sure that we're representing the physical system adequately to the model.
And I think with these emerging approaches, you're really getting the best of both worlds. You're getting the high throughput nature of sort of traditional QSAR and informatics, and you're getting the benefit of our insights into protein structure, as well as the decades of insights into just how well these different docking methods may be performing, and on their own, and why, and what tweaks help. I think it's a really interesting convergent time in the field.
Right, excellent, thanks. Right, so we've got one final question that's coming from Cheryl Arrowsmith, and this is to everybody who's participated in today's call. And the question is, what are the biggest needs moving the field forward? So, for example, more data for training, bigger computers, better algorithms. So I'll give each of you one thing that you will suggest, and you can, whoever wants to go first can go first. So don't hold back.
I've already typed an answer. So to me, the biggest problem is what we think it's true versus false versus in between. I've noticed that truth changes over time, and this disturbs me a lot. What we think we know today, I'll just pick on selective serotonin reuptake inhibitors. We now know they're far from selective. So there's lots of things that we thought we know, but we didn't. And that's really, if you want to build reliable machine learning models, you really need ground truth. Otherwise, garbage in, garbage out.
I totally agree, and we've seen that. I've spent 15 years doing DNA encoded library technology, and totally agree with Tudor. What you build in terms of your libraries, your target, and assessing your output. Rigorous assessment is absolutely critical.
Right, Ryan?
Yeah, I guess, I'll say something maybe, intentionally unexpected. one of the big problems for us is getting the compounds. You know, they do come in 5 to 6 weeks, but that's actually not fast enough to advance a project. So getting compounds quickly and at scale. We also have to work on scale, you know, because they're quite inexpensive, I think, for what you're getting, but we're still struggling with. We want to get 1,500 compounds, and we want to get them quickly, and we want to do them in cycles, and that's one of our big problems right now.
Great. Sounds
Yeah,
like
I agree with the sampling or the scaling question. That's kind of a limitation, that the cycle time is very long. I think we've talked about the AIMS collaboration project as sort of a delayed timescale active learning method. It takes a long time to get our predictions back and be able to say, these are things that we really learned something about, because these were predictions that we made and we committed to them, and now we're interested in seeing where we were right and where we were wrong. But we can actually build new models faster than we can get the data back for the quality, to evaluate the quality of the old one. So one of the limitations really is just the physical fact that assays take a long time, that compounds take a long time to make and ship. I was initially going to answer that question with data, but I think more specifically, I mean quality data on our specific type, the predictions that we specifically made, and getting that back at a fast time scale.
Yeah, and I agree with all those answers. Those were all excellent. And I would add, given that we do have these experimental and time limitations, we should be thinking about which experiments will add the most value to what we know and improve the models, not just try to find the next best, inhibitor for some new enzyme, but really pull back a little bit and think, where do we need to be exploring more rather than just exploiting?
Okay, thanks very much, everyone. Melanie, any last thoughts?
Yes, I would definitely agree with the previous presenters. And from my perspective, data quality is a major issue, and also the scalability of tools is very important to be able to extend all the tools and have still a nice performance and everything. Okay, great. So thank you very much to all our speakers and to all of those who've attended and asked questions. So it's been a really vibrant and exciting last 2 hours. The next Target 2035 webinar is on the 16th of February. It's on the topic of covalent ligand discovery and finding probes for challenging targets. So that sounds pretty intriguing. More details can be found, as you know, on the Target 2035 webpage. So I'd encourage everybody to go and look at those. And so with that, I'm going to call the session to a close. I'm sure if we were all in one room, we'd be applauding wildly, but you'll have to take it from us all as a sort of virtual thing. And great to see and hear everybody, and look forward to the next webinar. So thanks very much, Christina and colleagues, for organizing it.